Good afternoon. So I'm pleased to introduce Dr. Sadhana Jackson. She is a tenure track investigator at the National Institute of Health. For those who don't know what NIH or National Institute of Health is, they are our nation's medical research agency. They make important discoveries that can help improve our health and save lives. Dr. Jackson's research focuses on, she leads a major laboratory and clinical research focusing on characterizing the blood-brain barrier and blood tumor barrier in malignant gliomas. Her laboratory studies pertain to comprehensive evaluation of BBB and BTB biology by analysis of the structure, function, and expression with goals to transiently open both those barriers and improve drug delivery. So we'd like to have a warm welcome to Dr. Jackson. Thank you.
Thank you so much. Okay, I'm going to press this OK button. So thank you all so much for inviting me to Monell. To be honest, I didn't know that Monell existed. My research in my area is not in smell and senses. So I'm happy to tell you about the blood-brain barrier and the barriers in my life, and to be able to answer any questions you have in the research space and my role as a physician scientist. So as we start, maybe is it advances? Okay, I have no financial disclosures. I work for the National Institutes of Health, which is federal government, and I can't get paid on the outside, so no financial disclosures for me. This is cool. Please raise your hand if you play a musical instrument. Please keep your hand raised if playing a musical instrument comes easy for you. You just kind of have just a natural talent with it. I love it. Please raise your hand if you resonate with any of these pictures here in terms of being super organized and putting things in their correct places. And that gives you joy to have things in correct places. Please raise your hand if you can title yourself as a people person. Like, it's really easy for you to chit chat with them, easy to engage with others. You're extending your hand just as this fella is in the top left corner. So I showed you these pictures of these various situations and events, and these are kind of all things that we could describe as innate qualities about us, things that exist or belong or determined by factors that are there since birth, things that are just make you you. Yes, you play an instrument, but if it comes a little bit easier than others playing instrument, that then becomes your innate quality. That's you. That's just something that you were born with. And so I say that to say that I had, this is me at around 8 years of age, had an innate quality of just asking a whole bunch of questions and being super curious. And I love this quote by Ken Robinson that says, if you'd like to spark a curiosity in a child, they will learn without any further assistance. So I had that natural innate quality of curiosity as a young child that was just further kind of cultivated as a child from my parents and my family members and my teachers. And so I'm so grateful for that. And so that's one of the main reasons for why I'm here today, to talk to you about the blood, the brain, the barriers. So in talking about the blood, so my journey so far, because we're all on a journey, right? Mentorship and sponsorship and why that's so important. And because I'm a neuro-oncologist, I'll talk to you about what the blood-brain barrier is, what the blood tumor barrier is, and then some of our research of going bench to bedside, back to bench. And then to talk about barriers of influences of myself as a young Black woman in science and research in this world. So I love quotes. And so you'll see that I have varied quotes throughout this presentation. And so Bear Bryant said, there's a lot of blood, sweat, and guts between dreams and success. So remember that it's not an easy road, and that there are all these different bodily fluids that can come out as a consequence and as a productiveness of success. So my academic journey so far, so I'm glad that Patrice did not take the time and go through all the details of that, because then she would have stolen a little bit of my thunder. So I started with my interest in the NIH and just science in an undergraduate scholarship program called the UGSP. And my smile was good enough and my braces were good enough where they put me on some of their pamphlets to advertise for the program. But in essence, it was a program where they paid for my, NIH paid for my college education, my first 2 years, but I was had to sign paperwork that had for me to come and work at the NIH for the 2 years that they gave me scholarship support for. So I tell you that I now work at the NIH, but in essence, I got the job that I have now at the age of 17. So kind of a weird flex to have. But while I was at Hampton University, I was there for 4 years. I got a Bachelor of Science in Molecular Biology. And then along the way of me learning that I was a people person, so that third slide of raising the hand, I said, hey, go to medical school, even though I, really like science and research, and I had done I'd done laboratory research in college. I said, oh, but I could do clinical research and see patients and do laboratory research. Let me go to medical school. And so that's what I did. So another 4 years, I went to medical school, and that was in Norfolk, Virginia, which is very close to Hampton in Virginia. And then from there, I said, I really love taking care of children. I think they're cute. Even when they're sick, I feel like I have a good relatedness to them. I can be silly with them. And so I did a pediatric residency for 3 years in Orlando, Florida. And I loved it. And I was like, I love the additional challenge of caring for children who have cancer and caring for children who have blood disorders like sickle cell disease or thalassemia or clotting disorders. And so then that led me to Memphis, Tennessee, where I did a fellowship. So that was another 3 years. And then I said, you know what, that's not good enough in terms of medical school and pediatrics and cancer and blood disorders. What if I got another degree or another kind of training and I did neuro-oncology, so specifically children with brain cancer. And then I had an interest in how the drugs were getting into the brain. So I did a pharmacology or neuropharmacology fellowship. So that's a lot of training. You see my 2 kids up front. I still claim that I'm 31. They deny it. But yeah, so I did a lot of training to get to where I am now. And I still look 31, right? Yes, yes, yes. And then that led me back to the NIH. So just as she stated before, NIH stands for the National Institutes of Health. The National Institutes of Health really was created in 1887. It was a one-room laboratory in New York, Staten Island specifically. It got designated by Congress in 1930, and really it's the world's largest source of medical research funding. And so how you all may know the term NIH is that you know that NIH gives out a lot of money to different people. They give out grants so that people can do research outside of the NIH. But actually on the NIH campus in Bethesda, Maryland, it's composed of 27 different institutes. So the National Institutes of Health, and I work for one of those institutes, the National Institute of Neurological Disorders and Stroke. I also have a joint appointment with the National Cancer Institute. So I actually work in 2 institutes. But the overall mission of NIH is to seek fundamental knowledge about the nature and behavior of living systems and to apply that knowledge to optimize health and prevent or reduce illness for all people. So they have this big, lofty goal of not just giving out money to everybody, but to really just overall help the health of all people globally. So I've been there for about 9 years. And before I was there at the NIH, and even now, I have acquired various mentors throughout the time. And you can see all the different people that have helped to shape me to who I am today, and all those different places that I traveled to be able to get that specialized training. And so I want to pull these pictures up, just not to just show you these random people's faces, but to just show that it takes a village to really be able to make you into who you are today. So I know that you all can identify with various people who have helped to support you to be a part of this program and to be a part of the next programs. And so it's good for you to know who is on your side and who is your mentor, and the difference between a mentor and a sponsor. So the mentor will help you and say, oh, why don't you apply for this, or why don't you apply for that? Whereas a sponsor goes the next step, and he or she or they will not only say that you should apply for a certain program, but they'll actually advocate for you to be a part of that program. They'll bring you up in rooms, your name in rooms that you are not in. They'll nominate you for certain awards. So that's what you also need to aspire for and look for in your whole line of being successful, not just to have a mentor, but to have a sponsor. So I know it's kind of early, but it's good to think about these things now so that you know who's in your corner and who's not in your corner. And it's good to know, too, in the context of medicine and science, that it's not a very diverse field. What we see is a predominance of really white individuals being represented in the field of medicine. You can see all these different other ethnicities represented. And sadly, Black African-American females in medicine make up about 2% of the population of who is actually caring for patients. And you are looking at the only Black pediatric neuro-oncologist in all of the US. So I make up an even smaller percentage of that 2%. So we are doing better, I would say, in the US of improving the diversity in medicine, but it really plays a role in who then looks like and who you identify with for your sponsor and your mentors, and who's taking care of these patients to make sure that we're advancing care, okay, for everyone. And so I also, when I talk about mentorship and sponsorship, also like to be able to say that my peer mentors are awesome sponsors and mentors. So I'm sure maybe some of you know a few of these faces, but definitely Dr. Joseph, who is all in the research of senses and taste and smell specifically. But these people on this screen have been awesome advocates for me, and we've been awesome advocates for each other as it applies to research, as it applies to awards, as it applies to don't talk to that person. That person's probably not the best sponsor and mentor for you. So being able to navigate this world is just hard. So it may be the person who's sitting next to you that may be the best mentor and sponsor for you. So just recognize that somebody with gray hair doesn't need to be the only mentor and sponsor for your success. So the brain, fill your brain with giant dreams so it has no space for petty pursuits. Sometimes it can be fun to be petty. So just recognize that. And that this is just a quote. So there's that. So let me talk to you about the brain. So I am a pediatrician. I take care of children. Specifically, I take care of children with really aggressive brain tumors. And specifically, they're called high-grade gliomas. And then what I put in parentheses, all the different types of high-grade gliomas. Maybe you've heard in the news or heard in your research, glioblastoma. That's a really aggressive tumor. That's the tumor that John McCain had. That's the tumor that Beau Biden had. This is just a really aggressive tumor that after we give therapy, these patients do not live for long periods of time, even though we give every type of therapy we can for them. And for children, they do just as poorly as adults. In our lab and in our clinical research, we look at what happens over time, so time-dependent changes. We look at what therapies can really influence the blood-brain barrier as well as the glioma cells. And we look at the drug concentration, what actually gets to the brain, into the brain tumor of these patients. And the reason why this really makes a difference is because when we look at the treatment, a surgeon will go in and take out that ugly brain tumor. A radiation oncologist will shoot their beams and be able to kind of zap and fry that brain tumor. And then me as an oncologist will allow and write for them to get chemotherapy so that those drugs get to the brain tumor. So despite those three really aggressive type treatment options, the survival is really poor in these patients who are 0 to 19 years of age. Oh, you said that you were going to give me a pointer, but I don't see one up here. Maybe you can change this pointer option. Yeah. There's a pointer, here we go. So if you look at 24 months, so this is on the x-axis is time. I think I'm good. On the x-axis is time, and on the y-axis is how long or what the percent of survival is. You can see at 24 months, so 2 years, the survival is less than 50%. And that's regardless of the timing. So we've done a little bit better over time, but pretty much it is not improved despite surgery, radiation, and chemotherapy. And so the reason why is because these tumor cells are super, super smart. They grow really quickly. They grow really quietly. Not all the tumor cells grow at the same time. They don't all act alike. And they are really sneaky in the context that they can evade any of our treatments that are trying to get across the blood-brain barrier. So you'll have this ugly glioma cell that's growing, and it's next to just normal brain tissue, and it'll say, shh, don't tell anybody we're here. Don't let the T cells come in and destroy. Don't let any of the chemotherapy get there. So we're having to become really smarter than these cancer cells, which it makes very challenging. And so I like to be able to point to these two graphical illustrations. So when you think about an intact blood-brain barrier or blood tumor barrier, so everyone in this room has a closed blood-brain barrier. And that's good because we don't want toxins to get into our brain. We don't want to have headaches. We don't want to have bleeding. We don't want to have all these different kind of side effects that you can have from having your BBB be more open. So ours looks just like this, where you have these pink endothelial cells that are nice and tight together. They're kissing. And then if you look at this, this is our blood vessel. And so if we take a drug by mouth or we get a drug in our IV, these blue kind of circles serve as the drug of interest. So the drug is staying in the blood vessel. It's not going to the brain. Maybe it's going to other parts of the body, but it's not going to the brain. But look, you see that these peach colored cells are these glioma cells. So that means if we have therapy that needs to be killing these glioma cells, like I said, these glioma cells are saying, shh, don't tell anybody we're here. BBB needs to stay closed. But there are certain parts of the tumor where the BBB or blood tumor barrier is very disrupted. And those are the areas where you see a lot of tumor cells. And so we can actually do a good job of getting therapy to these areas, but we don't get it to these areas. So you say, well, what's the big deal? There's so few here when you look at all the different type of cells in the brain. Well, the issue comes because if you look at the core of the tumor, so the center area of the tumor, you have what we would call a very permeable or open BBB. So we can get therapy to those areas. So that's the disrupted area. But as you go further and further away from the core of the tumor, this animation is super slow, you can see that the BBB permeability is not as high. And so what happens is we do an awesome job of treating with radiation, chemotherapy, and surgery, this core and sometimes this peritumoral area. But when it gets to these distant site areas, because the tumor cells are just kind of hiding out, we see that these patients usually have regrowth of those tumors in that distant and peritumor site because chemotherapy is not getting there, radiation is not being thrown there, and surgery definitely is not being there. So when we look at an MRI, if we look at the patient's brain even grossly, it looks normal. So we're at a disadvantage in terms of being able to treat this distal and peritumoral site area, but we know that these tumors like to grow. So over time, we've given everything but the kitchen sink to the core and the peritumoral areas, but 6 to 12 months after we've given everything but the kitchen sink, we see in these distant site areas, the tumor starts to grow larger. These patients start to not do as well. And we don't have any other therapies to provide. So our research in the lab and in the clinic is more, how can we improve therapy in the very beginning in these distal and peritumor sites areas so that they have a disrupted BBB or BTB similar to the core, and then it goes back to normal, okay? So that's what our 3 aims are in terms of time-dependent changes, what happens in that peritumoral core and distal site area for aim 1, And then aim 2 is how can we treat drugs so that we not only mess up the growth of the tumor in those tumor cells, but also open the BBB. And then aim 3 is when we open the BBB, how much drug actually gets there? Because just because we open it doesn't mean that more drug is getting to that site. So I'll tell you a little bit about something that's going on with aim 2, or that we finished, where we went from bench to bedside, back to bench. So let me first tell you about a cardiac stress drug. So this is a drug that if you went to your doctor, you said, "Doc, I'm having some chest pain. I don't feel so good." The doctor would say, "Okay, well, let's do some evaluation of what your heart, the vasculature around your heart looks like. Maybe there's a blockage, maybe there's something smaller." And so they would have you walk on a treadmill and you do a cardiac stress test. If for some reason you were unable to walk on this treadmill, you would get this therapy called regadenoson. Regadenoson is an adenosine A2A receptor agonist. It works on the adenosine receptor. It causes increased cyclic AMP. It causes vasodilation. So that just means increased size or the width of your vessels around the heart. In addition to increasing the width of the vessels around your heart or causing vasodilation, it also causes vasodilation in your brain. And when you have vasodilation in your brain, you get a headache and you're like, "Oh, I don't feel so good." And so what they found is when they gave this FDA-approved drug for patients who are unable to walk on the treadmill to do a cardiac stress test, about 26% of those patients got headaches. And so that made a group out of Cornell in New York to say, if patients are getting headaches when they get this cardiac stress drug because of the vasodilation of the vessels around the heart, and there's likely vasodilation in the vessels in the brain, maybe that's causing the BBB to be disrupted. So what they did is they took rats and they took mice and they gave them this cardiac stress drug, and they wanted to know, are they opening the BBB when you give this drug that should be for the heart? And what they found is that when they gave this cardiac stress drug and a really big sugar protein called dextran, it was a 70 kilodalton sugar protein, they could see more of the sugar in the brain of these mice and these rats who had normal brains. They didn't have any tumor, they didn't have any disease. And they were like, "Oh, this is interesting." So they published that. We don't work with sugar. We more work with brain tumors. So we wanted to know if we gave this cardiac stress drug and we gave it with chemotherapy, could we see more of our chemotherapy go to the brain? And so we first looked at rats who did not have any brain tumor. And we found that if we gave them this cardiac stress drug called regadenoson, and we gave them with our chemotherapy called temozolomide. So you'll need to remember temozolomide is short TMZ. So you all know about the popular gossip blog and TV show TMZ. So you're not going to forget that. But we put TMZ with regadenoson, and we saw that we could see 60% more of TMZ in the brain. I don't know if you want more TMZ in the brain. There's a lot of gossip. But more TMZ in the brain of these rats that had no brain tumor. And then we saw that like 2 hours after we gave the drug. So this x-axis is time. The y-axis is the amount of temozolomide or TMZ in the brain. So we were like, "Oh, that's great. We could see more chemo in the brain. That means less chemo in the rest of the body." Because chemotherapy makes you tired. It makes your white blood cells go low. Sometimes it makes your red blood cells go low. So you really don't want chemotherapy everywhere. You want it to go where you want it to go. And we found that 6 hours after we give the TMZ and the regadenoson, it goes back down to baseline. So that's good. That means not too much stays in the brain. That means not too much is in the rest of the body. So we were like, "Oh, this is great. We get more to the brain of animals who have a normal intact BBB like you and I. What about if a patient had some level of disruption already because they had a brain tumor?" So after a few more studies, we said, well, why don't we use a tool called microdialysis so we can measure how much is getting in a human being? And so microdialysis is a cool way. It's a very small, thin plastic tubing that the neurosurgeon places at the time of surgery. And it allows us to be able to measure how much drug, after the patient is taking the drug orally by mouth or through the IV, how much of that drug is getting into the tumor. So this plastic tubing is connected to a pump, and at a very slow infusion rate, we give fluid into the brain, and then it allows the kind of device to kind of act as similar to a blood vessel, basically, and to allow us to capture that same fluid back, but allows us to measure how much of the chemotherapy or how much of the drug of interest is in the brain. And we can put these catheters in the peritumoral area or in the tumoral area. This is just an ideal situation that we would put it in the distant site area, but usually the neurosurgeon doesn't open up the brain so wide. They usually will open it up to only where the tumor is. So this is a cool tool where we're able to measure in real time how much drug is getting to the area. And so with that, on the x-axis, we're able to measure the drug timing and the concentration in the y-axis. So we look at brain to plasma. So how much drug is in the brain versus how much is in the rest of the body, the plasma. And we're able to look at varied drugs at a one-time period or over time. So the advantages of using something called microdialysis is that it allows us to be able to look over a long period of time. But in the past, and even currently, there are times where all we've been able to do is just get a little bit of tissue at the time of surgery. And that is good because we can get how much drug is there at that one period of time, but it also is a bad thing because we can only tell what is going on at that one period of time when the surgeon goes in and picks out that tumor. So to date, we've really used microdialysis catheters for patients who have traumatic brain injury and less in the brain tumor space. So in the brain tumor space, there's been a few drugs that we've looked at, like methotrexate, TMZ, our favorite, cisplatin, 5-FU, cytokines, different electrolytes. So again, it's a really cool tool. We can look at how much drug concentration at that over a certain period of time. Okay, so back to the cardiac stress drug regadenoson story. So we wanted to know if we give TMZ and this cardiac stress drug, do we get more drug to get into the brain? So we saw it in the rats who had no brain tumor. We wanted to know in patients who have brain tumor, do we see more temozolomide concentration? So we took adults that were either seen at the NIH or Johns Hopkins in Baltimore, which is really close. They all had the high-grade glioma, and they all needed to have either some kind of surgery to remove the tumor or some surgery to have some of the tumor taken out. Majority of the patients had already been given temozolomide or TMZ in the past, so we knew that they tolerated, they didn't have any allergies to the medicine. And this drug, this cardiac stress drug, interacts with caffeine. So we also had patients to not be able to drink any coffee around the time of their surgery. So these patients, I asked for them to get a cardiac stress drug, so their heart pumping really fast, and I didn't let them drink coffee after surgery. So I can't really say that these patients really liked me, but it was for the science that we needed to figure out whether or not this drug could increase more TMZ getting to the brain. So just as a review, patients came to the hospital, they had surgery, they had these catheters placed right after surgery. And then on day one, they took oral temozolomide. And then on day two, they took oral temozolomide again, but the second time they were given a push of the regadenoson, the cardiac stress drug. And then this catheter can be removed right at the bedside. They didn't have to go back to the OR for that. So by raise of hands, how many people think that we saw that 60% increase in the humans that had these brain tumors? Okay. Two of my friends said yes. Okay, so before I tell you the answer, these are just 3 demonstrations of where these catheters were placed. So it's really cool because these catheters have a gold tip at the end, and it allows us on CT to be able to see exactly where these catheters are placed. So we can see that 2 catheters were placed in this first patient, 1 catheter was placed in the second patient, and 2 catheters were placed in the 3rd patient. And the reason why I show that is because of where the catheters were placed. Because 1 catheter in the patient 1 and 1 catheter in patient 3 were placed in what we would say is BTB, or the blood-brain barrier is open. So you would think more drug to get to where the BBB is more open versus the BBB closed. And so what we found is we did not see that 60% increase that we saw in the rats. Only in patient 2, excuse me, and patient 5 did we see that increase, but it still wasn't 60%. On average, it was about 40-some%. So we said, well, what happened to the 60% we saw in the rats when we gave the TMZ and the regadenoson? So again, on the y-axis is the concentration, the x-axis is the time. In blue, you see temozolomide only, so that's that day 1. And in red, you see the combination of TMZ and regadenoson. And only in patient 2 did you see an increase for the red, and only in patient 5 did you see the increase in the red. So I'm like, oh no, this didn't work in all 5 of these patients. So sometimes things work, sometimes they don't. And we wanted to know, well, what about if we look in the areas where the BBB is open versus the BBB is closed? Again, we did not see a difference. When we looked at the plasma concentration, there was no difference, so that's good. We don't want more systemic TMZ everywhere. We want it to be here, but definitely there was no difference when we looked at BBB being closed versus BBB being open. So with these results, we said, okay, we started with the rats, then we went to the humans. What can we understand more if we go back to the rats? So bench to bedside, back to bench. And so I had a former postdoc that looked at regadenoson and TMZ in rats who actually had tumor. And we wanted to understand a little bit more about what was going on in the tissue for these patients, for these animals. And so we looked at the normal brain and we looked at the tumor. And we need to plug in this device. And we looked at the expression of a tight junction protein. And this tight junction protein is what keeps these endothelial cells together and keeps the BBB closed. And we found that when we give regadenoson,
I apologize.
no worries.
Put that out.
Mm-hmm. Okay. So we found that if we give regadenoson, we saw that we could have decreased expression of this protein that keeps these endothelial cells tight and together. So we're like, oh, these endothelial cells are not so tight together. And then also we found that if you look at the animal's tumor to plasma concentration of temozolomide here on the y-axis, you could see more temozolomide in the brain of rats who had brain tumors. So the endothelial cells were apart. That means more temozolomide was getting into the brain. And so we're like, oh, okay, well, what if we give temozolomide with regadenoson multiple times and not just a one-time point? We give it multiple times and see if we can increase the survival of these animals with brain tumors. So who here thinks that we increased survival when we gave the cardiac stress drug and the TMZ together? Okay, a few of my friends. No, we did not. We saw no difference. So you can see control here in black, TMZ alone in orange, and TMZ with regadenoson in blue. There's really no difference in their survival in the animals who receive the combo or just the treatment alone. We're like, ugh. So there's something and everything to be learned in our failures, and not really failures, but our hypothesis being proven wrong. And so our takeaways mainly for this project, being able to go from bench to bedside, back to bench, is that timing is everything. So the timing of the BBB and the BTB disruption really matters when you think about when to give the regadenoson in the context of the TMZ. The brain endothelial disruption alone. So we kept saying that regadenoson is affecting these endothelial cells, But if you remember from that previous graphical illustration, I showed you all these other cells that make up the brain. So we have the astrocytes, the pericytes, the microglia. Those, we don't know how they're being affected by this cardiac stress drug, but for the most part, they aren't being impacted as much because this is more of a brain endothelial type treatment. And then understanding what's happening with the concentrations is really important. So we were able to do that with the microdialysis catheter. And so we're advancing the field and learning more about these brain tumors, and yet and still we get results that are contrary to our hypothesis, which happens, right? That's how science is. And so what we've really started to do with our AIM-1, AIM-2 is understanding what happens over time in that core peritumoral and distant site area, and look at what's happening in the cell expression and the progression of the disease. We're looking at different drugs, not just regadenoson anymore. We're finished with regadenoson. I don't even want to see that drug anymore. We're looking at other agents that can open the BBB and improve drug to be able to kill these tumor cells, to disrupt these tight junctions. This is what I'm showing here. These are tight junctions that keep these endothelial cells kissing. And we looked or published an agent that could disrupt the tight junctions. We're also looking at another agent that can mess up the tight junctions and also kill the tumor cells. And that's here to show that we would want more therapy to go to these peach-colored tumor cells. And then always looking at the drug concentration with microdialysis, wanting to know how much drug gets to the spongy cortex, so that's normal brain, versus the midline of the brain. And we can get tumors in all these different locations, but you can tell just from this picture alone that the architecture of this part of the brain is significantly different than the spongy architecture of this part of the brain. So as you can imagine, when tumors grow in the midline of the brain, these patients do horribly. We give everything to them and they still do horribly. So one of the last quotes that I place here is from your director and president of Monell Center. I enjoy bringing diverse groups of scientists together to help solve problems and drive innovation. We can do so much together, so much more together. And it's so true because we come together and we're like, I think I know the answer. I have an hypothesis and this is maybe what it is. And sometimes things come at you and you don't even think about having to formulate a question or a response to them. And so in terms of things coming to you and you not being able to have any idea about that, not to say my family came at me, but I love my family and they are my support system. And you can see a few of them in the audience today. My husband, my children, and this is just a show of some of my family members. Sadly, we lost my mom in 2021 to metastatic breast cancer. So you'll see this beautiful picture of her, us celebrating her at her 70th birthday. And then one of her next birthdays, and she was not here on this heavenly earth. And so it takes a lot to be able to practice medicine and be able to be this great mom and wife and friend, and community support is everything to be able to be successful in this world. So know who's there on your side at all times and who you can trust and who you cannot trust, because life does come at you fast. And we were all home at the time that we saw these sad, senseless murders in the streets, on the TV. And it came a time where many of us at the NIH, and even not NIH related amongst family, had conversations on what is it for us to do and say about the injustice in the world. And I found myself speaking to my husband and speaking to friends, and it's like, oh, I'm just a scientist, or I'm just a mother. Like, what am I supposed to do? How am I supposed to play my role in equity and justice and inclusion? And so interesting enough, there were a few of us at the NIH that got together, virtually, of course, in 2020. And we said, let's have an open letter that we would send to the NIH director, who was then Francis Collins, and tell him and leadership about the injustices at the NIH and how it's not really an equitable place here. in how people are treated and how people are being responded to, and just equity in pay. And let's write an open letter and see how that goes. Interesting enough, NIH has a history of being called out. We wouldn't have been the first ones ever in NIH history to basically point out some of discrimination within the agency. These are a few pictures from our NIH kind of newsletter and newspaper that have shown in the past that there have been discrimination and unfair practices at the NIH. Even people outside of the NIH, people funded by the NIH, have called for the NIH to be more equitable in who we give our monies to. And they see and saw that there was a lot of groupthink. So this graphical illustration is awesome because it shows, okay, these are the people that want to get funded. And so what happens is the people in orange are the ones that are deciding who's getting the money. And if the people weren't thinking all the same, groupthink, then they were being pushed off to the side. And so the people who are being funded are all looking alike, thinking alike. And so what that they're saying in terms of NIH racial funding equity is that there need to be more ladders placed so there's equity in who gets funded, and so that not everyone is thinking the same, and that there's more innovation and creativity. So there's been many papers published to say, NIH, you're not doing a great job of being equitable about who's getting the money, and how can you say that you really want to advance science and research if everybody is thinking the same? So we, as a group of about 10, 12, 15 of us, came together and wrote a letter on July 22nd, 2020. And we titled ourselves ACRE, or 8 Changes for Racial Equity. And we wrote this open letter to Francis Collins. And we sent him asking for 8 changes. The first change was publicly and explicitly identify diversity inclusion as anti-racism as a top NIH priority, to track and publicly report NIH workforce data annually, to improve representation of Black, Indigenous, and Latinx individuals in the NIH community, to implement an annual workplace climate survey to address all forms of harassment and discrimination, to develop required anti-racism and anti-discrimination training for all NIH employees, to require and incentivize leadership to be active participants in diversity, equity, and inclusion, to implement a wage equity plan so that everybody is being paid the same amount with regard to their training and their certifications. And they had recently done an anti-harassment kind of campaign around the hashtag #MeToo. And they said harassment does not work here. And we asked them to add in racism, discrimination, and harassment don't work here. So we sent this letter to the director of NIH, and we all kind of sat on our hands for about 8 days. I was like, okay, I'm a physician. There's some hospitals down the street. I could probably get a job there. I could probably be a consultant. Like, you know, NIH isn't everything. And in 8 days, Dr. Francis Collins responded, and he said, I agree with you. There is a lot of discrimination. There are a lot of inequities at the NIH. We do need to do better. And you can see from all these varied headlines, he came out. He apologized for structural racism in the biomedical research field. He vowed for change. They started a whole bunch of committees around how do we improve equity in the NIH and who we fund, and how do we make this place more equitable. And so I'm grateful to be one of the change agents that helped to spur these conversations and help leadership think through what needs to be done so that everyone feels comfortable. This is a screenshot of the Ending Structural Racism page at the NIH that came about after this open letter was placed. And then many of us who helped to write this open letter and champion change helped in serving as leaders of what they call the Unite Committee, or UNITE, or a different stand for an acronym of things around structural racism changing and improving at the NIH and those that we fund. In addition to becoming a co-chair of the T committee, so that's more on communication, I raised my hand and I said, now what about all of the artwork that's on these walls and these hallways that don't make me feel welcome as a young female Black scientist here at the NIH? I'm seeing a lot of older white males. So how do we usher in the new generation who don't all look like white males? And so they said, okay, so since you raised your hand, you can then start to help with the campaign to change the artwork. And so that's what we did. So I became the lead of a project called the Recognition Project, where we helped to put about more artwork on the walls that reflect who we serve, which is the global sphere, and who we would want to recruit more of to work at the NIH. And then scattered amongst the different images, so this is a picture of me, are various quotes that we had to have NIH library, of course, to reference and make sure that they were correct from various leaders within science and outside of science. And so the overall slogan for Unite is Together We're Stronger. And they did these beautiful, our medical illustrationists did these beautiful graphical illustrations of varied people who work at the NIH, and even some celebrities to date that they've added in. And we have people from various walks of life. We have people who are so grateful to have more of a diversity on these walls for the people who are coming in, for the people who are thinking about coming in. And I recently received an email from a summer student, and I'll read it to you. So, dear Dr. Jackson, I hope this email finds you well. My name is blah, blah, blah. I am currently an undergraduate summer student in NIAID, so that's one of the 27 ICs, who is studying neuroscience. I visited the NIH and saw your picture on the wall in the lobby and was immediately interested in talking to you, not only about your wisdom in neuro-oncology, but your experience being a Black female doctor who is also involved in research. I'm particularly interested in learning from your journey, the challenges you've encountered, and the strategies you've employed to overcome them. I'm reaching out to request a meeting with you to discuss your experiences and insight further sometime next week, or perhaps later this summer. I am flexible regarding the time and the format. Thank you. I look forward to hearing from you. So I felt so full reading this email that I received in 2024. Now, those pictures went up in 2022, but it worked right how we wanted it to work. We want summer students to be able to walk by these pictures and like, oh, I see myself reflected. Oh, I feel so bold. I'm going to cold email Dr. Jackson and figure out about her journey. So I met with her and she's just lovely. And so I told her, I was like, I used your letter. I took your name out of it, but I used your letter in my presentation. And she's grateful and I'm grateful. And we still have more work to do. So the barriers. Remember that problems are always just solutions in progress. Now, this is me. So you trademark me, right? So our lab really is focused on therapeutic targeting of the blood tumor barrier and malignant gliomas. So we look at time-dependent changes, we look at dual role therapies, we look at pharmacology. I love what I do. I love who I work with. I love to be able to share with you all things that I do in the lab with the patients and all throughout globally. These are just various things that I've been involved with because I work at NIH and because I don't. So I'm grateful for your time. And to remember, embrace your innate talents. We all raised our hands about those. Remember, it's both challenging and rewarding to remain committed to your aspirations and find strength in your vulnerability while being dedicated to being a change agent in this world. So thank you, and thank you. Any questions? Yes.
I was just wondering about this during your presentation. When you described the blood-brain barrier, were there any concerns about like, because like the whole purpose of it is to prevent like toxins or even like a poly drug or essentially multiple drugs. Did you ever think of that? And if this is to make it to like a clinical setting, what would you do about that kind of problem?
Yeah, so I get this question often. So it's like, oh, you're opening up the BBB, maybe that's going to cause more problems, right? Or infections. So we have not seen that to happen. And the intent is to open it for a short period of time so that it goes back to normal. Interesting enough, our BBB becomes open every night as we sleep and we dream. So we know physiologically that's just a normal state that happens. So when you're studying and it's like, oh, I got to go to sleep because I got to like make sure I retain all this information. You need to go to sleep, be in that REM, be in that non-REM, be in that deep sleep so you can make sure that that BBB gets open. It absorbs that data. It's able to wash out those toxins in the clearing. So we're in essence attempting to mimic what normally happens at nighttime but for a short period of time. And so that's why I had that one terminology of timing really matters, because if we're using regadenoson or some other agent, we're going to want to open it up, but we're going to want to have whatever our therapy, TMZ, whatever, is there right at the time of opening. So it gets in and then it closes.
I didn't know that.
Yeah.
I have another question about the blood-brain barrier research. So I'm actually working on a lab that does a lot of antidote at schools. And I was wondering if you ever thought about doing research on something that would not require you opening blood-brain barrier, but rather have drug investment.
Yeah, so there's a lot of different labs that have looked at nanoparticles, nanodrugs, exosomes, extracellular vesicles to try to increase drug delivery. That's not our research area, but yes, it's definitely a budding field. It's been hard to get those agents FDA approved. So it takes a long time. So in the meantime, we're going to keep moving to try to transiently open things up and see what kind of targets that we can hit to be able to say, we're going to kill these tumor cells as well as open the BBB to bring more therapy in. But definitely that's a great field. So if your interest is that, I think that you probably will have a long career in that